Hello everyone, this is Dr. Marcon, and this is the discussion for Lab 23 on white blood cells. So with this lab, we're doing a total white blood cell count, sort of similar in method to when we took the total red blood cell count. However, this time, because there are a smaller number of white blood cells, we are counting larger squares, and we will probably get a smaller cell count compared to red blood cells. So we want to count the four squares, the four larger squares labeled A, B, C, and D. We can probably expect to find anywhere between 25 to 50 white blood cells within each square. So again, when we took a sample of blood, we placed it in a tube that had a solution that basically lysed or got rid of all other cells besides the white blood cells. We then used a hemocytometer and placed the plasma or the fluid into the hemocytometer again, and we are counting the number of cells within these large squares. So for easy calculation, let's say that each large square had 50 white blood cells. So 50 in A, 50 in B, 50 in C, 50 in D. So that gives a total number of 200 cells. So we then take this number and multiply by 50. This adjusts for dilution as well as volume counted. So we know that this is influenced by dilution accuracy and sample homogeneity. And this will give us total number of white blood cells in thousands per microliter of blood. So the normal number for the total white blood cell count is approximately 5,000 to 10,000 white blood cells. So taking our original number, we had 10,000 when we multiplied 50 and 200. Leukopenia is a condition where you have less than 5,000 white blood cells, and it's always pathological. Usually we see these in patients that are immunocompromised. So whenever you see the suffix penia, just think a decrease in the amount of cells. When you see the word cytosis, cytosis means an increase in the amount of cells. So leukocytosis is a total white blood cell count more than 10,000. And usually we want to see normal leukocytosis, especially in the presence of an infection. However, leukocytosis can be bad in certain cancers such as leukemia. A leukemia can be considered myeloid if it originates in the bone marrow, and a lymphoid leukemia if it originates in lymph tissue. So these are the types of leukocytes that we are looking at, and white blood cells can be categorized into two groups, either being granulocytes or agranulocytes, depending upon the presence of cytoplasmic granules. So granulocytes do have these cytoplasmic granules, and there are three subtypes to the granulocytes. Those are the neutrophils, the eosinophils, and basophils, and these names are usually based on the type of staining that you will see of these cytoplasmic granules. Neutrophils having a neutral stain of their cytoplasmic granules. Eosinophils, you'll usually see a dark red staining of the granules, and basophils, you will see a dark blue or purple staining of the granules, and usually this dark blue staining of the granules will occlude or prevent you from seeing the bilobe nucleus of the basophil. Agranulocytes have no cytoplasmic granules, and you have two subtypes. The smallest subtype being the lymphocyte. When I talk about lymphocytes, I usually talk about it having a fried egg appearance, so a very large nucleus and a small rim of cytoplasm. And then the monocytes being the largest of all the white blood cells. It's a very large cell having a kidney bean shaped nucleus. And my favorite description of a basophil, because of its dark staining granules, I like to say that it has a chocolate chip cookie appearance. Now make sure you are familiar with the basic morphological features that distinguish the leukocytes, and we discussed them a lot in the lecture on blood. So this slide just reminding you of the prevalence of the specific cells or specific white blood cells. The mnemonic again is never let monkeys eat bananas. So the N standing for the neutrophils being the most prevalent, and we know that neutrophils make up about 60 to 70% of your white blood cell count. The next most numerous is the L, which stands for lymphocytes. So lymphocytes being the next most prominent, composing about 20 to 25% of the white blood cells in your white blood cell count. You'll definitely run into them if you ever properly do a blood smear. The next most prevalent being the monocytes. Again, these very large cells being the third most prominent. Percentages about 3 to 8% of the total white blood cell count. Next is the eosinophils. Eosinophils not being very prominent. You'll probably see about 2 to 4% within your differential WBC count, and then the least prevalent being the basophils. You're lucky if you run into them. In my anatomy class, I actually offer extra credit points if they are able to find, if my students are actually able to find basophils, because they are definitely very rare. And if you see an increase in the number of basophils, this is usually due to some underlying pathological condition. So again, we use the mnemonic never let monkeys eat bananas to help us remember the prevalence of the different white blood cells in the differential white blood cell count. So make sure you are aware of the prevalence of these cells, as well as morphological features that help distinguish the cells from one another. So these physical characteristics that help you distinguish the cells. So just a basic review of the histology from anatomy if you took anatomy. And now there's a discussion on the white blood cells.